You can find this presentation on Zenodo if you go to this link, and I'll walk you a little bit through this data set and also the science behind it, because that's always a little complicated with the data sets alone. Like, what is it about? So you would need the narrative paper. And so I want to motivate you also a little bit for the science behind this data set. And then I want to go to the meta level, because I think that's really interesting today to have the discussion with you. Like, what did we do and is it really good enough? And so some of you might know this poster. It's the metabolic pathways poster, which is pretty big and still just a tiny snapshot of what you can expect in a normal biochemical cell. And if we zoom into that part at the top, and if we zoom in further, then still the reaction I investigated, and Niels as well, and other students, it's still not there. But because we're just a super tiny footnote on metabolism here. But we think it's still a very, very interesting reaction. And why that? Because of DNA and the nucleotides it consists of. And now for a little refresher. So we have the DNA, we have 4 bases coupled to sugar, and you have the 4 nucleobases on top, and then linked to sugars. And this is then a nucleoside and a nucleobase on top. So the reaction we looked at is this: you take a nucleoside to the very left, deoxythymidine, and then you add phosphate and an enzyme. And what it does is it detaches the base and couples a phosphate. And this is what we did. And if you do this now in the lab, you have an enzyme reaction in an Eppendorf tube. You take a small sample and you quench it in a sodium hydroxide solution. You transfer it to a 96 well plate to 1 well, put it in a plate reader for 30 seconds. Then you get the UV-Vis absorption graphs. You do some in silico data processing, and then you can go and work to the conversions that you actually see in the chemical reaction. And now this was already new, because before we always had to use HPLC, which took 15 minutes a run. And now we are down to 30 seconds, and it's also cheaper because we don't use the solvents. And so how can you now, from the graphs on the top, work out what it means, this strange absorption? And that's something we had to rediscover. If you vary the pH and you vary it in a solution of the nucleobase, you get this shift kind of behavior. So there is another species popping up with a different absorption spectrum. And if you do the same to the nucleoside, it doesn't vary its form, just a little bit the absorption. And so you can use that difference to then rework what was the concentration of nucleobase and nucleoside in your solution. And because that's what we're interested in that reaction, it works really, really well here. And yeah, like very short background, because this is now actually about the data set. And the data set we want to work on, now you understood, okay, we have UV-Vis absorption spectra, we want to go and back calculate what was the conversion in this sample. So you would need a program which Niels and I wrote mostly, then a metadata file, which we found really, really helpful to have it in one file, the metadata about your spectra, and then a human to check, because actually the computer makes mistakes sometimes. So it doesn't make mistakes because computers don't make mistakes, but you end in a wrong local minimum. If you want to check the data set itself, it's also online on Zenodo. like very general purpose repository, and you can search there. Yeah, you can find it if you want. What's in there? So the most important part from my perspective, a license giving you the right to reuse our data without asking us again explicitly, because in principle it would have been copyrighted, probably. Then a README file, so you understand what we actually did there, what the files are about, the metadata files I talked about, the scanned lab journal pages of Niels, and then the actual data in proprietary format and in an open format. So this is UV-Vis absorption. And so to give you an impression, so we have here 3 files, and they all now link with each other. And now I would say this is getting a little unfair, because you would now need a lot of background knowledge, what we actually meant here. And a machine is very unlikely to figure this out. But if you start from one of our metadata files, you can see in description in the column, we put this NK for Niels Krausch design 1. If you go to the other file, you see, okay, this is the condition name. It has this data file. So you go and look up in that data file, and you see well number D5, and then was a specific time point. And then the computer now, of course, can do that and link it all up, treat the data, and give us the conversions we're interested in. And of course, you could do this all with Excel and manually, but we found it really helpful. And then Felix followed up on this, and he produced like 10,000 data points. So he saved a lot of time with not doing it with Excel, but having it automated. So now, because it's on Zenodo, for example, you can also go to Google Dataset Search and find it. But I would argue this is not really findability in the FAIR sense. A small side note, we found out that there is a lot of those equilibrium constants, and actually a lot of them not known for those nucleoside phosphorylase reactions. So we also started a new database on those for enzyme catalyzed chemical reactions. And yeah, if you do the same and you're interested in contributing your equilibrium constants, you're very much invited to visit this Google Groups. It's mostly a discussion group right now, and we are curating some data from the literature. And now I would like to take you to the meta level, which is it fair? Is it good for chemistry? And like, what did we see right now here? And I would argue there's a current state of the art. So if we take 100 random journal articles from your field of interest, and you would look what people do, this is current state of the art. If you now look in that field, what are the best examples of good research data management? I would name that best practice. And then we have a vision, and this vision is fair data. And I like to illustrate it on this Transformers thing, where you can make a robot out of a car, which looks strange. So you have the vision of this robot and you have a car. Like, what are you going to do next? And now you have this vision, you have to figure out a good next step, and always go one step ahead. And then you see, okay, it's really possible. But we are really on like level 1 or 2. So we are just opening the car a little bit right now. And our vision is really far. But we have to work out next steps right now. And a little self-critique, how could this data set actually have been better? And what did I wish for? What can you do? So none of this data set, in this data set, there are no machine understandable names. Like, we don't have inches. We didn't use UPAC names. If you look for UV-Vis absorption, but you call it differently, Like, I don't know, you don't use a hyphen, you use a slash, you don't say absorption spectra, but you call it somehow different. You wouldn't ever find our data set. And it shouldn't be like this. So with the thymidine, for example, usually the definition is thymidine is always without the oxo group. So it's thymidine, and we call it deoxythymidine to make it very clear, but it's actually like, it's the same name or the same thing, but different names. You would want to find it always, no matter how you call it and how I called it. It would have been nice if we could have submitted our UV-Vis spectra to a repository. We didn't find one at the time, at least. The results of our reactions, like the yields and equilibrium constants, so we try to make up or create a new database right now, but we didn't find one. So this is always a pain. And then a digital electronic lab notebook, we didn't have one. So we scanned the pages. And then we did some computations. Should we link to it? Then again, the question for all of this and more, which falls in our data set, is there an open standard? And often we didn't find one. So we as a community did not yet agree on a standard way to do it. And I believe as long as we don't agree, it's hard to take a next step. And then personally, what did I wish for? When I was preparing the data set to submit it to Zenodo, and great would have been a checklist, because I always felt very unsure if I didn't forget anything. Then an example of what's good, because all I knew was data available on request. So I didn't know, like, an example to copy. That would have been nice. And then someone to talk to. Now NFDI for Chem, I could write you an email, I guess. And then it would be great if someone would check my files, like, are they complete for at least an outsider? And that could be peer review, like in the traditional journal, but right now it's mostly not happening. Then I didn't have any pressure to deposit my data. So that's like anti-motivation, because we were doing extra work nobody requested us to do. And then looking at the audience, I thought interesting could be, what can you do to make the next step happen? And I would argue, if you're a journal editor, you can make sure somehow, with a lot of pressure and long time horizons, to have in your journals a data availability requirement, and to have it openly available, and that data available on request is not something to write in a paper. And if you have that requirement in the journal policy, that it's also enforced, because even if it's in there, from what I've seen, it's not always enforced. If you're an author of a paper, today, I would argue, just take all of your data and dump it on Zenodo as it is, because that's very easy. And if you want to take an extra mile, do it. But if you don't, dump it. I'm happy for any data that's on Zenodo and that doesn't get lost in the course of time. And that's already it from the content side. I would like to acknowledge all of the team, and yeah, I didn't do this alone. If you have any questions, always feel free to reach out to me. There are some bonus points in the slides online, like proposals I would do, or like looking from now back in time, what would have been good and what are easy things you can start doing today. And if you have the time, I would be happy for some feedback for this talk. And you can find it on that small QR code or this tiny CC feedback, Robert. Yeah, that's it. Thanks.